So in tertiary structure now, tertiary structure, what we have is that those coils that we have in the secondary shape, they then start to fold onto themselves, right? So the coils start to fold onto itself to give a 3D shape. I'm going to use a hose to demonstrate that to you. So if you look at this hose, if we look at the spirals on it, we call it the secondary shape. So when this starts to fold up on itself now, fold in and fold all over, we could call this a tertiary structure shape. But if we go back out and have just the helical shape like this straight, then that would be a secondary shape. But anytime you see it start to fold up on itself now and twist up, if I had a telephone cord, it'd be better, then we call that the tertiary shape. Just to be clear, now we say tertiary structure is when we have the coils start to fold on themselves and give a 3D shape. Now that 3D shape is held together by hydrogen bonds. Now, well, not exactly by hydrogen bonds, but in addition to hydrogen bonds, we have ionic bonds, disulfide bonds, and hydrophobic interactions. But these three, ionic, disulfide, and hydrophobic interactions, they occur between the R groups of the different amino acids. So if you remember, please recall that we said the general structure of the amino acid is of such: you have an amino group, central carbon, and then carboxyl group. The top, the R there is where we have the variable group, or the side chain. Now, depending on what molecule sits here, we can have ionic interactions, we can have disulfide bonds, and hydrophobic interaction. Now, please note that each polypeptide chain is coiled around a small group of atoms with iron in the center. That group is called an heme group. And when we talk about a heme group, we only referring now from the tertiary structure. So when you reach the tertiary structure, you have that 3D shape and all the different interactions that happen with this side group or the R groups that fold it. They always have a group of atoms. At the center of that group of atoms, we have an iron group. We have an iron molecule, and that heap of 3D shape is bonded around this heme group here. That heme group, although a part of the polypeptide chain, it is going to be called a prosthetic group. So let me clear up that part, because I think maybe somebody don't understand that. So when what I'm saying about a prosthetic group is that we all know that proteins are made up of amino acids which form polypeptide chains. But I'm telling you now that when the polypeptide chain is in the tertiary structure, it will coil around a group of atoms which have an iron in the middle of them, and that group, we call it the heme group, right? This actually helps the polypeptide chain in its function, but it is not really, say, a basic unit of protein. It's just a heme group. So we call them a prosthetic group because it's like an addition to the polypeptide chain to help it to function properly. All right, so the prosthetic group is really the heme group, which has a iron at the center of the little group of atoms. All right, so I'm going to try to do a little visual explanation of what we mean by tertiary structure and identifying the heme group. So did a little drawing here, and at the center of the drawing, that green box with that red dot would represent my heme group. At the center of my heme group is the iron atom. Alright, so the iron is at the center and the red dot, and the green box represent the small group of atoms surrounding that iron in the center. So these two put together makes my heme group. Now, the polypeptide chain is folded upon itself, and that folding looks kind of untidy, but that's what we mean by the helical and the beta and everything coil up together, the hydrophobic interaction and everything coil up together and wrap it around this heme group. This is a polypeptide chain in its tertiary structure. Now, alright, so since you catch that one to understand what the polypeptide chain is in its tertiary structure, let's look at hemoglobin for a second. Hemoglobin is a protein molecule that is found in your red blood cell, the pigment that makes your red blood cells red. And it's a protein molecule. It has 4 heme groups, and if you have 4 heme groups, that means you're going to have 4 iron molecules at the center of those heme groups. If you have 4 heme groups, then you're also going to have 4 polypeptide chains, because remember over here I just show you 1 heme group have 1 polypeptide chain, wrap up, coil up, twist up in a tertiary structure wrapped around it. Good. Alright, so we have the 4 polypeptide chains. Now, in hemoglobin, the 4 polypeptide chains, we call 2 of those chains alpha chains and 2 of those chains beta chains. Good. Now, the 2 alpha chains and the 2 beta chains curl into balls, making hemoglobin globular. Good. Globular. I want to stop it right there now to pick it up at the quaternary structure. Alright, so looking at hemoglobin again, we said that it have 4 heme groups, 4 iron, 4 polypeptide chains. We said 2 of those chains are alpha chains, 2 of them are beta chains. Now, in order to get the complete structure of hemoglobin, those 2 alpha chains and 2 beta chains are linked together and they curl into a ball, balls really. And they make hemoglobin a globular protein. So you will come across the term globular protein. Another example of a globular protein would be enzymes. All right. So what we mean by a quaternary structure now. So you had to learn about the hemoglobin in order to understand quaternary structure. Quaternary structure refers to the 4th level structure or 4th level organization. All right. So if I do a visual representation now of what I mean by a tertiary structure, what you'd see, this is an example of hemoglobin. So you can see I have 2 into 1, 2, 3, 4 heme groups with 4 polypeptide chains. I put the beta polypeptide chains in blue and the alpha in black. So these 4 polypeptide chains put together make 1 hemoglobin molecule. So 1 hemoglobin molecule have 4 iron at its center, 4 heme groups. Good. So because you have these polypeptide chains bond together, once you have more than 1 polypeptide chain bond together, then we shift from tertiary structure to quaternary structure. So quaternary structure really is just the fact that you have more than 1 polypeptide chain bonded to a heme group. Once you have more than 1, so you don't have to be 4. So just to reiterate, for tertiary structure is just the level, is just a 4th level organization. Good. So it means, therefore, if I have only these 2 beta chains bonded here, this would still be quaternary structure because I have more than 1 polypeptide chain linked together. If I had the 4, it's still quaternary structure. If I want to go back down to tertiary structure alone, once I have 1 polypeptide chain bonding upon itself around the heme group, this is the tertiary structure. But if I have more than 1 of these polypeptide chain bonded to each other, then we go on to quaternary structure. Good. When we have them more than 1 bonded together and they curl up like a ball, we call that globular protein. Example of a globular protein is hemoglobin and also enzymes.